People relate bacteria with diseases. In some cases that's true, but that's a really small percent of bacteria. Astonishingly, less than 1% of different bacteria types make us diseases. Most bacteria are helpful. In fact, humans depend on these bacteria to help them with most of their biological functions. On the other hand, most viruses are harmful, but a really small percent of viruses are harmless, or sort of harmless. In this video specifically, we'll be comparing bacteria and viruses in terms of the structure, size, shape, way of infection, and most common diseases. Viruses range from 5 to 300 nanometers, while bacteria range from 0.5 to 5.0 micrometers. It's worth noting that a micrometer means 6 decimal points and a nanometer means 9 decimal points. It's now obvious that bacteria are way bigger than viruses. However, if you have noticed, a small percent of viruses are bigger than bacteria. That's when the size of the virus is the maximum point and the size of the bacteria is somehow below that point. Concerning the shape, bacteria has 4 shapes. Round and spherical bacteria, aka cocci, rod-shaped bacteria, aka bacilli, coma bacteria, aka rhibrios, and finally corkscrew bacteria, aka spirochetes. On the other hand, viruses have 4 shapes: filamentous viruses, aka icosahedral viruses, envelope viruses, isometric viruses, and finally head and tail viruses, aka bacteriophages. Bacteria shapes has subcategories called genera. Pairs of cocci are called diplococci, rows or chains are called streptococci, grape-likes are called staphylococci, 8 or more cocci are called saccharinae, and 4 cocci are called tetrads. Others have similar genera, while corkscrew bacteria, aka spirochetes, have Spirochetaceae, Brachyspiriciae, and Lithospiriciae. Now with the viruses. Filamentous viruses are long and slender. And note, most of the icosahedral viruses are plant viruses, like the TMV tuberculosis mosaic virus. Envelope viruses have membranes surrounding the capsids. Most of them are annual viruses, like the HIV. Isometric viruses are roughly spherical, like the polioviruses and the herpes. Last shape is the bacteriophages, or the head and tail viruses. It's obvious from the name that they attack bacteria. Their head is similar to the icosahedral viruses, and their tails are similar to the isometric. Ways of infection is a very complex topic to talk about, but I'll try to give you a brief summary. Bacteria can be transferred to the host through various ways, like air, water, food, and living vectors. The principal modes of infection are contact, airborne, droplet, vectors, and vehicle. Before the bacterial invasion begins, bacteria needs to find the right host cell. To do that, they use pili, which are hair-like structures that stick out of the bacterium. At the end of the pili is a little patch. When the patch finds the right cell type, it sticks to it. Connecting with the host, bacteria have little hooks called adhesions. The adhesions bind specific protein components on the host cell. After pulling itself with the pilus, or the singular form of pili, the adhesion helps to bring the bacterium closer to the host cell. Now each bacteria spreads its toxin material to the host cell and makes it infected. For sure, the body tries to fight off the bacteria with a process called phagocytosis, but that's not our topic today. On the other hand, viruses have 2 ways of infecting the cell. Let's have nicknames for them: the long cycle for the lysogenic cycle and the short cycle for the lytic cycle. Basically, both of them are the same, but the lysogenic cycle has some extra steps, which is why it's the long one. First step is the attachment of the virus to the host cell, in a similar way to the bacterial stickness spike. Second, the viral DNA enters the nucleus of the host cell, and now the separation begins. Both cycles have these 2 steps, though let's leave the lysogenic cycle for a while now. After the viral DNA enters the host cell, the cell makes more viral DNA and proteins. The virus completes its form, and finally the new viruses leave the host cell to infect the other cells, which is the lytic cycle. Now back with the lysogenic cycle. After the viral DNA enters the bacterial cell, the viral DNA becomes part of the bacterial chromosome. Lets it replicate more and more till the provirus leaves the bacterial cell and then back to the last 3 steps of the lytic cycle. Bacteria are prokaryotic cells, but before I start, I'll now explain organelles that are present in both the eukaryotic and the prokaryotic cells. Okay, so let's go. Bacteria have nucleoids, which have the main DNA, plasmids, which have extra DNA, usually for protection and antibiotic resistance, capsids, which is an outside layer for extra protection and helps keep sticking, flagellum, which helps with movement, pili, will help with sticking and other functions, and finally ribosomes, cell wall, cytoplasm, and plasma membranes. Virus cells aren't living cells, so they won't have any organelles. However, they do have an envelope protein and an envelope which make up the protein coat. Inside there's the viral genome, which can be made up of DNA or RNA, nucleocapsids, and at the end there's the viral tegument, which helps the viral DNA in replication. Most common viral diseases include measles, rubella, chickenpox, Zeola, smallpox, fish disease, flu, HIV, and herpes. While the most common bacterial diseases include leptospirosis, pneumonia, cholera, botulism, MRSA infection, E. coli infection, meningitis, gonorrhea, and bubonic plague.
You know, it's kind of fun watching your own videos. Of course I made that, but it looks cool, right? Okay, whatever. So, I'm thanking you for watching my video, and I hope you enjoyed, and that's not copyright free, so do not do that, other than if I give you permission, obviously. Okay, bye!